So chapter 20, we're talking about spectrophotometers. So the idea of this chapter is we're taking what we learned in chapter 18, which is about the fundamentals of spectrophotometry. How do we relate these spectroscopic measurements to concentrations? How do we relate this to analytical information that we're interested in? But of course, to be able to do these measurements, we have to build a spectrophotometer. We have to be able to actually construct the thing. And so the idea behind this chapter and the way I personally view this is there are many different techniques we could look at. So spectroscopy is, of course, something I do personally, and so this is the one that I like talking about the most. But we could talk about other things like mass spectrometry or chromatography or other types of analytical techniques that are used. And for each of these, we could have a whole discussion on how to actually construct these in different parts. And so just think of this as a specific deep dive into spectrophotometers, but I hope what you're able to get out of this is sort of the general idea and sense of these techniques that we use require a lot of work to be able to construct something, and a lot of decisions that need to be made, different types of materials that we can use. And so we're going to do this with one specific type of analytical technique and application here with spectrophotometers, but we could do the same thing for many others. And for those of you who have had or will have instrumental analysis, that's basically what that entire class is about, is understanding, it's building on what we talk about in this class and understanding how we can construct different sorts of instruments, how they work, and how we can actually put them together. And so this is really at the heart of a lot of analytical chemistry is understanding what's possible and then trying to expand what's possible with different new materials and techniques and pieces of equipment that we can use. So to get started, let's kind of outline what we're going to talk about. And the best way to outline it for a spectrophotometer is just to look at the diagram of a spectrophotometer and kind of think of the different parts that are involved. So this image I have here is a slightly modified diagram from the spectrophotometer we talked about in chapter 18. We've added a few extra things that are common in most modern spectrophotometers. So of course we have our light source. This will be the first thing that we talk about in this chapter. Then we have monochromator, and then we have our cuvette. And this is really where the main difference is that many modern spectrophotometers actually have two different cuvette holders. So there's one where we put our sample and another one we put our reference. And the idea here is that instead of needing to switch out your blank for your sample and needing to do that every time you take a measurement, you can just have one blank, and the spectrophotometer is set up with a motor or with a rotating mirror, which is a beam chopper, that splits the light between the sample cuvette about half the time and the reference cuvette about half the time. And so you're able to measure the light between both of those. And so those both go onto a detector, which we'll talk about as well. And then the signal from the detector is amplified by some sort of electrical circuit sent to a computer or display to actually give us the reading. So what we're going to talk about in this chapter is we'll be talking about each of these components. So talking about light sources, there'll be a video on us, this first video. We're talking about monochromators, how we construct those and sort of the things we have to think about in terms of the design, different types of detectors. And then for infrared spectrometers, the most common type doesn't follow this sort of same pattern that we've been talking about. We'll talk about Fourier transform infrared spectrometers. And then the last topic we'll cover is dealing with noise. And defining what we mean by noise here, not actual acoustic noise, but noise in our measurements. And so I just included here a picture from your textbook of, if we take the box off of the top of our spectrometer, this is sort of what it'll look like. We'll have some sort of light source over here, and we'll often have more than one light source. So this has deuterium and tungsten halogen lamps, and we'll talk about those in just a minute. And then the monochromator is down here. We have a grating that's part of that, a couple of mirrors that are part of it, and some slits. And then that light gets sent into our two cells here, right, our reference and sample cells. And then we have a detector over here. That's in this particular case, it's a photomultiplier tube. And something that looks like this actually has all this complexity underneath this sort of white box where we just put in the cuvette, we get a reading out.